So Momo, I want to now take just a step back. A customer gets the Viome test. They send them the most precious thing they have, their poop, to you. Now I want to see the journey of this poop into the things that they see, what food to eat. Can you share this background of what happens to my poop that goes in there?
The poopy journey. Okay, so yeah, this is an interesting thing. So first what happens is that poop is preserved in our special preservative that freezes this RNA molecule the way exactly it was when it was produced. That way, during shipment, there's no degradation of these RNA molecules. So the sample arrives at our labs, and the first step that we do is we crack open all of the microbes. So the technical term for that is cell lysis. So we physically crack open every single microbe in the sample, and then that's how they spill their RNA content into the solution. And now we take those RNAs, which there are billions of from every single one of those organisms, and we
Hold for a second. The tiny poop that I sent you had billions of those things?
Many, many billions, yes. The molecules are incredibly small, so billions of them. So we don't want to actually sequence all of them because 99% of them are non-informative. They're called ribosomal RNAs, and they're just basically occupying the space there, and we don't want to sequence them because we already know their sequences, and so they're non-informative. So we actually fish them out physically from the sample, and now that 1% that remains, we sequence that 1%. So we identify the actual sequence of A's, C's, G's, and U's in this case. So RNA has a U instead of a T, but it's the equivalent of a T in DNA. And we sequence all of those, and we actually sequence 10 million of those molecules for each stool sample that we get from our customer. 10 million.
You mean 10 million times?
We sequence 10 million molecules. Okay. Different molecules randomly from the pool, and they represent the microbial community that lived in the poop.
Wow.
And so now we have very, very fancy bioinformatic analysis that analyzes every single one of those molecules individually. So we not only identify which organism that molecule came from, but we know which gene it came from and what function was encoded by that gene. So not only do we know which organisms are there at the strain level, we now can identify and quantify the gene expression of all the microbes in the poop down to the level of 1 in a million. So if a gene is expressed at the level of 1 in a million or higher, we can actually identify and quantify its function.
That is unbelievable. 1 in a million, right? Wow. So keep going. So what happens to my poop now?
So now that we've identified which genes are expressed by the microbes, the work continues in putting those genes together into pathways. So remember, I just talked to you about how a microbe can use some food molecules from our food, right, and convert them into, let's say, pro-inflammatory biochemicals. That's called a pathway, an enzymatic pathway. So it's a system of enzymes which come from genes that convert that food molecule into a pro-inflammatory molecule that causes inflammation. And so we actually put together several genes into a pathway, and we quantify the activity of that pathway. And that's how we measure. We don't infer, and we don't have models for that. We actually measure the activity of these microbes, and that's how we have this inflammatory score that tells every customer how high their gut microbiome-induced inflammation is.
And then continue. So what happened? How do I get my recommendations now?
Okay, so now let's go back to that simplified example. So that one microbe that is able to produce both good and bad compounds, pro-inflammatory and anti-inflammatory, we now know exactly which substrates, which foods it's using to produce pro-inflammatory compounds, and our computer model will immediately place those foods into minimize and avoid. And then that same microbe, remember, same exact microbe can produce anti-inflammatory compounds, and we now know which foods it uses in order to produce those chemicals, and we want it to produce those chemicals, so we want to feed it those foods. And so now foods containing those particular molecules are actually placed into the enjoy and indulge list, which means that we want to feed the microbiome those foods so that it can produce anti-inflammatory biochemicals. And that's how we fine-tune the effects of gut microbiome on our physiology. We give it exactly the molecular foods that it needs in order to produce beneficial and not to produce detrimental chemicals.
And that's how we know that when I say don't eat these food, we tell you why you shouldn't be eating this food. And if we tell you to eat a certain food, we tell you why you should be eating certain food.